So we will wait for 5 more minutes.
Okay, sure.
Good morning, sir.
Good morning, ma'am. Good morning.
Yeah, after long back. Maybe forgotten. I know you, sir. Long back, when Ram Reddy, sir, from USA, regarding
Yes, yes.
calling, that time we met.
Yeah. This is a long time.
Yeah, after long time, sir.
Wonderful. So happy.
Thank you so much. See you today.
Very happy here to be in touch
Yeah.
with you. Yes.
Okay.
Okay, sir. Shall we start the session, sir?
Yes, please.
Okay. Good morning, everyone, and welcome to Day 4, Session 9 of Faculty Development Program on Innovative Strategies in Drug Discovery and Therapeutics, conducted by RBVR Women's College of Pharmacy. It's my pleasure to have you all here today. My name is Sajida Afreen, Assistant Professor from Pharmaceutical Chemistry Department. I would like to introduce our eminent presenter for today's session, Professor B. Jairam sir. Sir has completed his doctorate degree from City University of New York, USA in 1986 under the mentorship of Professor David L. Beveridge. Sir has then worked as postdoctoral fellow in Columbia University in New York, USA from 1987 to 1988 with mentorship of Professor Barry Hornick. Sir has also worked as senior research associate at Wesleyan University, Connecticut, United States from 1989 to 1990 with Professor Beveridge. In 1990, Professor Jairam Sir joined IIT Delhi and worked as faculty from 1990 to 2019 and as emeritus professor at IIT Delhi from 2019 to 2023. Sir has been head of chemistry department at IIT Delhi. He has been founder coordinator of Kusuma School of Biological Sciences at IIT Delhi. Sir have been founder coordinator of supercomputing facility for bioinformatics and computational biology at IIT Delhi. Professor Jairam Sir served as a mentor of Program Advisory Committee of Department of Science and Technology, Government of India for Physical Chemistry, Organic Chemistry, Biochemistry, Biophysics and Molecular Biology. Sir have been a member of Bioinformatics Task Force of Department of Biotechnology, India from 1998 to 2007 and 2013 to 2018. Sir have been a member of Bioinformatics Working Group of Department of Information Technology and Bioinformatics Task Force of ICMR. Sir have been a member of IUPAB National Committee from 2008 to 2011 and DST FIST Committee for Chemical Sciences from 2009 to 2010. Sir have been a Vice President of Indian Biophysical Society from 2007 to 2009, Chairman of DBT's Committee on Promotion and Popular. Sir have been a Chairman of DBT in Bioinformatics National Certification Exam in 2018. Sir is currently co-chair in Data Management Committee DBT from 2024 up till 2027. Sir was a guest editor for special issue of journal Proteins and Proteomics in September 2017, which released covering of Bioinformatics Conference organized by SCF Bio celebrating its 15th anniversary of its inauguration. Sir have also been co-editor of journal Journal of Bioscience on nucleic acids in disease and disorder.
Rajita, can we move
Sir, have
on? Can we move on? I think that is okay. Thank you very much. Kind introduction. Let's get. Yeah.
okay. Bit of it because that is the most, I think, highest achievement of your such good softwares like Chem Genome, Bhagirath, and Sanjeevni for you for that. And now I thank
Now
you.
the major thank you very much. Major challenge is to know. Let me share my screen. Then we start.
Okay.
Are you?
It is visible, sir. ఒకసారి కాల్లో చూడండి అమ్మా.
Yes, ma'am.
Sorry, I think you can call him on phone.
Okay, ma'am. Okay, ma'am.
Maybe some net issues or any other issue.
Yes, ma'am. Yes, ma'am. We are calling.
Yeah, join. Okay. Okay, I was trying to share my screen and suddenly something happened. Okay. Let me do it quickly before.
Yes, sir.
So, are you able to see my screen, please?
Not right, you know. Yes, sir. Yes,
Visible? Is
sir.
it okay? Okay, before the network creates problem, let me start. Thank you very much again. Thank you all. Thank you, Vijay Bhargavi and Rajat. Very nice to talk to you all. Hello? Hello. Just let me know when suddenly it stops, right? Can I continue?
Yeah, it is. Okay, sir.
Okay, thank you. So I'll share with you a big long journey from genome all the way to hit molecules in silico. This is created by about 25 PhD students of mine and several hundred project students at least, or several number of them. So I'm sure you'll enjoy the journey, a country path I call it, and your students may contribute to the science goes through a hypothesis generation, validation, etc. So don't forget the top path. While we are busy applying AI everywhere, right, machine learning and artificial intelligence everywhere with the big data, ensure that you also create hypothesis. That is how you push the science, right? Newton's law, first law, second law, third law. Such things can happen only with the top, not with the bottom. With the bottom, you get a prediction, right? You don't know how it happened, but you get a right prediction, good prediction, etc. So keep both in mind. If you are in a hurry to get a product useful to society or a drug molecule, maybe ML AI can get you there, but simultaneously focus on using the data, how to generate a hypothesis and how to validate it and eventually create a product, etc. Right. So those are things that I wanted to share with you. Now, big data, lots of molecules. And the biggest challenge people have these days is that we have 4.5 billion ready to dock compounds. So many small molecules are available, yet the diseases seem to proliferate. We are not able to control the disease growth. How do we stop these viral infections or bacterial infections or other kinds of diseases and more deadly ones like cancer, etc. Right? So the dream plan we had, this was way back in 2000. At that time, we started to decide to create a supercomputing facility dedicated to drug discoveries, software development, and so on. And we also thought we should make it freely available to the entire Indian community. That was the motivation. And so we sketched out a pathway. The pathway was that you have a genome. From genome, you identify a gene, and from gene, coding for a protein. The protein sequence can be written easily thanks to our Gobind Khorana's codon concept. Once you have the sequence, you build a 3D structure. Then you're looking for a binding pocket. And once you have binding pocket done, you do docking, scoring, etc. And finally find a molecule. And once you have that molecule, you send it to a panel of doctors who will prescribe the dosage and ensure that it's safe for the patient. Patient then takes the medicine and is running enjoying. So the idea is 10 a.m. the disease is reported, 5 p.m. the drug is delivered through Amazon or Zomato or one of those, right? So that is the overall plan. Maybe 20 years from now this will be a reality, but today we are still working on the details as to what are the details, what are the challenges involved to ensure that someday we'll have a disease-free planet. So that's my goal today, to convince you in the next 30-40 minutes that this is feasible, right? And this is not wild dream. This can be done, right? And of course, now what we hear from WHO and others is not only the drugs part, you also have to worry about the antimicrobial resistance, the AMR. Because of the AMR, And since there is a discovery void of antibiotics, we may run into some serious issues unless we discover some new antibiotics. So there is there's a matter of urgency that we have to come up with some new antibiotics. Whatever we have, the bacteria started developing resistance to these existing antibiotics. So one more reason why we should get serious about this drug discovery part. Now, sketching quickly some milestones in the drug discovery. Some of you already know this, but just to share with you some ideas on what's happening, right? As to the first Nobel in drug discovery, quote unquote, happened in 1939 for frontosil. That was an antibacterial. Remember, 1939 to 45 was the Second World War time. People came out of the First War, and then there was Second World War, and most soldiers were dying due to bacterial infections, and the frontosil, then came penicillin. So these were all, at that time, there was a big hurry to produce penicillin in huge amounts, right? So these were the people who were responsible, and I'm sure all of you know how Fleming got there, right? Serendipitous discovery, accidental discoveries. And this lady, Hodgkin, and seems to have now come up with a structure, a so-called beta-lactam ring of the penicillin molecule. She also did the B12 structure. She got the Nobel. And streptomycin was another against tuberculosis. This was in 1988. So now what you see here is that there is a molecule, and that seems to cure a disease, right? But how do you get to this molecule is a big issue. And this lady, you too, some of you know, has been working on artemisinin for a very, very long time. And this is from Chinese traditional medicine. And this is a very effective molecule against malaria. Right? So we got the Nobel for 2015. And then came the question, how do you go about doing drug discovery, right? So that was a principle initiated by these three scientists. They said, focus on a rational approach, focus on a biochemical pathway or a physiological process, and try to address that issue. If these things have gone bizarre, then you want inhibitors, right? So that biochemical physiological process has to be the underlying, not random testing of various molecules, because there is no end to it. You keep testing millions and billions, and you cannot do this on humans, obviously, right? People may die. So toxicity could be a serious issue. So you need a rational approach. So the idea of that drug discovery has to be rational. This started coming about in 1980s, and 1988 Nobel went to this idea, right? And all of you know now, Venki Ramakrishnan. And why is this so significant? Well, as according to Ada Yonath, this particular ribosome of bacteria for which they developed the crystal structure, and they got the Nobel Prize. So that is different from that of humans. Now remember, ribosome, what does it do? It takes mRNA, pairs with tRNA, and then creates proteins, right? So that's a very essential part of life. Proteins are the working horses, right? Workhorses of our life, and therefore bacteria gets disabled. Protein synthesis in bacteria is disabled if you are able to block the ribosome of bacteria. So the structure has enabled. So now we are now talking of not just rational design, but also structure-based drug designs, right? That's become very popular after that. Now coming back to this idea that if you have billions of molecules available, why is it that only 2,500 drugs are developed in the last 80 years? So that is surprising to me, very surprising, because we have so many fantastic chemists, organic chemists in particular, some geniuses really. In spite of this, diseases are on the increase, and the number of drugs which are approved by FDA, FDA stands for Food and Drug Administration of USA. If they put a stamp on your molecule, you can sell it anywhere in the world, right? So this is surprising. In spite of the increase in genomic and proteomic data, somehow the pipeline is drying up. So that's very surprising, And of course, we can blame it on the cost. The cost, of course, is 14 years and $2.6 billion. This is back. Now they've increased it to $3 billion, which is very, very expensive, obviously, right? So our expectation then is that if you do a computer-aided drug discovery, you can probably cut down the cost and time in drug discovery, right? And if you take care of toxicity in particular, then you probably are going to accelerate drug discovery. So that's the issue that I will address. So now back to our dream plan. How do we achieve this idea? 10 a.m. disease reported 5 p.m. drug is delivered at your doorstep. So let's say we have genome information, right? You could have any one of them. You may have just the genome information, or you may say, I already know the gene. I have a protein sequence, right? And or alternatively say, I also have the protein structure, but I don't know the binding pockets, active site, etc. Or you might also say, well, I know binding pocket as well. I just need a molecule in this particular pocket. So you have several areas, several entry points in this pathway that I'm showing. You could start with the genome, you could start with the protein sequence, a DNA sequence, or a protein sequence, or a protein structure with a binding pocket identified, and then of course you can create a molecule, right? So that is the general idea here. Now I keep calling them as Chem Genome, Bhagirath, and Sanjeevani. So these are three popular softwares created by us, and also I use this abbreviation SCF Bio all the time. SCF Bio stands for our lab at IIT Delhi Supercomputing Facility, etc. And all the softwares that I'm mentioning are freely available. You can just log into this site, or you just have to do a Google search, or just click on this URL and you're going there. You can use any of the softwares for free. Now let us see, does it work? The big challenge. Now, as you can see, this got published in 2020. I felt so good. We started this whole thing in 2002 when we created this SCF Bio, created 20 years and several PhDs. Finally, we have the entire pipeline worked out. So Chem Genome to Bhagirath to active site finder, then rapid screening of molecules, parallel, and filters. Now I'll tell you more about these things as we go on. Now, I call this entire pathway as a Dhanvantari pathway. How does it work? So one of my friends in the School of Biological Sciences, he is world's expert on hepatitis B virus. So he says, can you give me a molecule against HPV that I can test it? So he has all the experimental setup, and he is asking me, do your computer-aided drug design, CADD, and give me a molecule. I will test it for you, right? I told him very simple, give me 14 years and $2.6 billion, I'll get your molecule. No, more seriously, I told him just 3 hours. Give me 1 day, maybe. 3 hours is too short. 1 day, I'll get your molecule. Now, I asked him, what do we know about it? He says something about this HPV virus is already known, ORFs, open reading frames, and corresponding protein products, viral polymerase, etc. So he was asking me, don't focus on the polymerase because everybody is developing drugs against this viral polymerase. Focus on other proteins. So that was his challenge, one challenge. The second challenge was, don't give me a nucleoside analog. Now, what is a nucleoside? AGCT are nucleic acid bases. You put a sugar, it becomes a nucleoside. You attach a phosphate, it becomes a nucleotide, right? So we are looking at, most of the drugs are nucleosides with a sugar attached to one of the base-like molecules. They are not exactly AGCT, they are modified bases with the sugar attached, so that virus confuses with them as AGCT and incorporates using the polymerase, incorporates in the RNA, DNA, and therefore the virus gets clear, right? That is a general philosophy of this drug discovery here, as this as an inhibitor. But he was asking me, because of the resistance that's developed against nucleoside analogs and also polymerase, so give me a new target as well as a new molecule. How do we do it? Now, I request you, for those of you who have not used these softwares before or not heard of these softwares before, just to make a note of them, and then you can try it out yourself. Whatever I'm doing is repeatable, you can do it yourself. So Chem Genome is the software for virus genome. Virus genome is available at NCBI. Remember, this NCBI is the National Center for Biotechnology Information, USA. And you can log into this site, just walk into the site, and you see several genomes, including human genome. Viral genome is small. You can easily download it onto your laptop. Once you have the laptop, the viral genome on your laptop, then cut and paste that into this Chem Genome. Chem Genome is a front end for our software for genome annotation. What it does is to identify 5 protein coding regions. And then I was looking at all the proteins, but again, my friend said, don't do polymerase. So he asked me, why not gene 3, etc. So I said, okay, no issue. We could have taken any one of them. So that's how the protein looks like. Methionine, glutamate. This is a single letter amino acid code, asparagine, isoleucine, etc. Now I take this and then put it into this Bhagirath software, which does this protein structure prediction. Now, some of you may have heard of AlphaFold. Right? This AlphaFold is supposedly doing a miracles in terms of structure prediction. So if you are a fan of AlphaFold, feel free to use it. Once you have this chem genome giving you the sequence, you can submit it to AlphaFold to get a structure. Alternatively, there are other softwares, including this Rosetta, etc. Bagheera is one of them. And so you have a structure. Once you have the structure, then you start looking for binding pockets. Where are the binding pockets on this protein? Are active, so-called active sites, right? So for that, I use my Sanjeevani Active Site Finder software. It identifies 10 binding pockets. Now comes the challenge. Since we have those millions of molecules available, can we now do something about them? In fact, we also have simultaneously developed a BIM called Bioactivity Information of Medicinal Plants, Indian medicinal plants, which has now about 1 lakh compounds from various plants in India. So that is another database. So why not test all the molecules in these databases? You cannot do this experimentally in a laboratory because you'll be wasting too much of chemicals and a lot of money, time, and so forth. So let the computer do the job for you. How does it do it? So this RSPD, which is supposed to scan millions of compounds in a matter of minutes, it does it in a matter of minutes. So really seriously, yes, try it out. RSPD of Sanjeevani, I now scan these millions of compounds again as these binding pockets. And then what the RSPD does is to give me some 150 molecules based on some cutoff. So I set minus 10 kilocalories as cutoff. So then I take all those 150 molecules. But beyond that, the next step is to go for atomic level docking. So then I do my van der Waals, electrostatic calculation, Coulomb interaction, solvation, desolvation, etc. Those kind of detailed chemistry-based, statistical mechanics-based, delta G naught. Remember delta G naught minus RT ln K, that equation. So I'm using that to get my free energy of binding at the atomic level. This is atomic level. So the previous one was virtual docking. Now this is the actual atomic level docking. The previous one, of course, here does millions in a matter of minutes. So this paradox, of course, takes about, it works on 8 processors, takes about 20 minutes or so on the 8 processors, and gives you some 5 structures, which are really good from a binding point of view. And then beyond this, what we recommend from our experience is that do MD simulations so that you have flexibility taken care, you have solvent taken care, etc. So finally, we have about 5 molecules recommended from this entire exercise for HBV. And then I told my friend, take one of these molecules. I'm sure they should work. And that was the idea. So my job is done. Remember, I was mentioning to him that within a day, I'll give you a molecule. Right? So from genome, you go all the way up to the molecule prediction. And what are the major steps? Genome annotation through chem genome, structure prediction through Bagheera, and then Sanjeevani active site finding, and then virtual docking through our RSPD, then Paradox, then MD simulations. At the end, you are ready with your prediction. Does it work? It's a big challenge, right? Now, interestingly, this molecule, this was the first publication, 2021, and my friend Vivek, of course, did a lot of experimental validations, and it got published. So this is Ruchika was my student, Kiritika was his student, and these are my collaborators who got the structures, etc. Vivek Perumal is a collaborator, right? So it does really work, this entire pathway. In fact, Vivek found out that this molecule that we suggested works against drug-resistant variations of the hepatitis B virus as well. So he published a couple of papers on this in the last 2 years. So that's wonderful to see that this entire pathway seems to work. Of course, we have several success stories, not just this one, but this essentially it tells you how to go about this pathway. Now, quickly, so those are the right people who made it possible, right? All those PhDs who did the job. Now, some challenges. I'll mention to you now some challenges and some more things that you can do or you can inspire the next generation to do. So I'll skip some of these elementary slides, but just to highlight the history part. The first RNA genome came in 76, bacterial genome 95, eukaryotic 96, human genome 2001. This was that NIH and Craig Venter's contribution, right? But the big challenge that I have is, what is the meaning of the CAT, GAC, CGT, etc.? So, of course, what you typically do is to compare this with what is already known using homology, using some similarity. Then we say, well, this must be a gene, it's not a gene. It may be something else. It may be a control element or box or promoter, etc. So reading DNA is still not feasible. That's the question. So how do we read DNA? What is the language of DNA is the question that's still bothering me, bothering everybody, not just me. Interestingly, Craig Senker is the same person. The DNA, he did the DNA sequencing here, and earlier he did the protein sequencing. Got 2 Nobel Prizes. Right? One of the 5 who got 2 Nobel Prizes. People. Now back to the genome and proteome, etc. My question is that you couldn't have predicted what's the meaning of TAG, CCC, etc. unless I gave you this reference definition, chromosome X, etc., chromosome Y, and so forth. So that's the basic question that we need to address. And how do we handle this question? Well, we have gene prediction software, several of them. Most of them use interesting machine learning or training methodologies. So you have some sparse experimental data and you're training your data to do prediction, right? And then what happens with that approach is that it works well for the organism on which you train. Like you get 97% accuracy, but you go to another organism, it drops, sensitivity drops very badly, accuracies drop very badly. So there is no universal model. So the other thing that I was mentioning to you is not just sensitivity, specificity, they're not 100%, but the methodology requires training. You need to do a training with experimental data. You need the experimental data. So this is not like what Watson and Crick would have done. Remember Watson, Crick, methodology, A versus TAG pairs with C, right? There's no training involved. It just happens. There's a nice chemical interaction between A and T, G, and C. So unless you somehow bring in that chemistry into your model, it's not very clear whether you can create a universal model, which works for bacteria, prokaryotes, eukaryotes, humans, plant. So how do we handle this? So that got us thinking about not ignoring the structures of adenine, thymine, etc., and also focusing on the major groove, minor groove, and so on. If you need a universal model, you must factor in the chemical nature of bases. You cannot just go into sequence space. You have to bring in some structural information as well into your model, right? And remember, DNA is a very flexible molecule. So these are all the various base pair. This is a base pair. This is a base pair step, right? There are one base pair and another base pair. So there are 2 base pairs here. So this is a step. Whereas these are base pairs, so they can do shear, buckle, stretch, propeller twist, etc. A lot of things can happen. DNA is a very dynamic molecule, right? And it also depends on the sequence. Not only the bases, you also have the sugar phosphate backbone. Backbone also shows lots of variability. There are variations here, right? And you have 6 dihedral angles and 5 sugar pucker and a chi, that is sin and tie type orientation of the bases and so forth. So just remember that DNA is a dynamic molecule. Now, what we decided was, let us go for a universal model. Whether we succeed or not is a different story. Don't do training. No training, no experimental inputs into it. Go with some kind of an ab initio model. How do we do this? Well, we said, like, why not we focus on, just like Watson and Crick said, just focus on this base pairing, A versus T, 2 hydrogen bonds, G versus C, 3 hydrogen bonds. So hydrogen bonds should play a role. If you have lots of GCs here, you'll have lots of hydrogen bonds, meaning that this molecule will be very stiff. If you have less number of hydrogen bonds like AT or TA, then you will have flexible DNA, right? This may be important from promoters. Remember, promoters bend the DNA. So if you have lots of ATs, you can bend them. If you have too many GCs, you cannot bend them, right? Promoters won't be able to bind. And so they cannot recruit polymerase and then protein expression and so on. So this must be playing a role. That was one proposal, hypothesis. The second hypothesis is that this is called stacking. One base pair on top of another base pair, this is called stacking. Stacking interactions should play a role. What we are learning from MD simulations is that if you have a step like this, so this is a thymine, which is single ring, I call it pyrimidine. This is G, purine, double ring, right? So look at this double ring, right? So pyrimidine to purine, this is, I call it the YPR step, P for phosphate. YPR step, pyrimidine to purine, is a very flexible step. Purine to purine is not that flexible. So if you want a very flexible kind of DNA, you want to put in more YPR type of thing, single ring, double ring kind of thing. For example, single ring and a double ring. Here's a CA, right? In the 5', 3' direction, of course, right? So that's another part that's called stacking. interactions. Now, third thing that we can think of was what may be happening if you are creating a universal marvel. Remember, our job is focus on the language of DNA and not worry too much about whether it's a prokaryote or eukaryote. Is it a rice genome or a human genome? You don't want to do training. That was the intention. Now, how do we go about it? Now, here, there are these grooves here, major groove, this is a minor groove. Proteins are binding in these major and minor grooves, and they are dictating protein expression, all kinds of things. Whether gene should be turned on, off, proteins are deciding. So proteins know the language of DNA. We don't seem to know still, right? That was there. So we have three parameters now. That's not complicated. So hydrogen bond, stacking, and third is a protein nucleic acid parameter. In the absence of protein, solvent water is all over the place, right? So solvation energy. So using these three, we can easily derive these numbers from MD simulations. But if you don't have this kind of MD expertise, don't worry about it. You can always do a back of the envelope calculation, pencil, paper. Each hydrogen bond is 2 calories. So if there are 2 hydrogen bonds, there should be 4 kilocalories, etc. You can make some hypothesis and then create a table like this. And then once you have a table, if I give you a sequence, what do? Suppose I give you a long gene sequence. Well, all you have to do is to take the dinucleotide, keep adding them up, A, C, G, etc., etc. So that is simple addition. Simple calculator will do the job. We don't need any computer anymore, right? And one thing that I don't want to spend too much time on, but I want to mention, however, is the question of Babel. People have said that Babel explains it all. We have 64 codons, but we have only 20 amino acids. And Francis Crick said third base is wobbling, and therefore you don't have 64 amino acids, right? That was his explanation for Babel. But then, if third base is unimportant, then we have only 16 possibilities. 4 times 4, only 16 amino acids, but we don't have 16. We don't have 64. We have somewhere in between. When does it bubble? When does it not bubble? That was the question that bothered me in particular. I said then, maybe there is a rule. There is a rule, I call it as a conjugate rule, that A acts like the opposite C and U acts like the opposite of G. Or in the context of DNA, instead of U, you call it as a T. Thymine opposite acts like the opposite of G. So whatever GG does, first base, second base, TT does, it's a conjugate as does the opposite. So if GG codes for one amino acid, its conjugate TT should code for more than one amino acid. Now you see this is true. Similarly, if CG codes for one amino acid, its conjugate should be AT. AT codes for more than one amino acid. In fact, this rule works without exception. And you see very few rules in biology. And why is this so? Why does it work? Well, that's because of the hydrogen bonding and stacking impact, right? So T makes 2 hydrogen bonds. What are these hydrogen bonds, by the way? These are mRNA, tRNA. mRNA binding to tRNA in the bed of ribosome machinery. And it's not TT, then you call it a UUC, etc. Instead of T, you will have U, right, for RNA. So that is essentially what is explaining the degeneracy in the genetic code and also Babel. So I'm bringing this up just to tell you that whatever we hypothesized earlier, that stacking hydrogen bonding should play a role, that seems to, in fact, work well when it comes to Babel hypothesis explanation. So we have complicated it enough now. Let's 3-parameter model. And does it work? Well, what we do then is to take a sequence, add up all these values, create a unit vector, and ask where is the orientation of the unit vector. We tested it on 900 prokaryotic genome. So most of the methodologies, as you've seen, require training, right? Training on a particular species. We don't need to do any training here, right? And so this is MD-based, molecular dynamics parameter-based methodology for genome annotation, identifying genes. So each dot here, blue dot, happens to be a gene. So you have the entire organism on a sphere here. Right? Red ones are frame-shifted genes, which are not supposed to be coding for proteins. So that was the idea. Now, once you have this method worked out, easy to write a software. So my students, are good. In fact, in my lab, what I do is for each team, I put one bioinformatician, one chemistry PhD student, and one computer guy, MCA, etc. So all the 3 or 4 of them are working together to create a software. Right? So that is typically the strategy that I follow for each of the softwares. So reading faster format, then ORFs, and eventually the hydrogen bond stacking solvation vector. Is it above, below? If it's below, then it's not a gene. Above, it's a gene. That was the idea. So this is the server that I was mentioning to you. Now we have the ChemGenome 3.0. So all you have to do is to cut and paste your sequence here, and then just run ChemGenome, and you get your information. Right. Now comparing this ChemGenome with other softwares which are available. Remember, now I am using Arabidopsis thaliana, which is one of the eukaryotes of plants. Right. So I've tested it earlier on prokaryotes. Now I'm doing it on thaliana. Well, it's not bad at all. Sensitivity, specificity are not bad at all with a simple 3-parameter model. So it seems to work quite well. But you may say, why is it not 1.0, 100%, right? That would mean maybe we need a 4-parameter model. So it's still not at a Watson-Crick level. Probably some more tinkering is required to get to that point. So not only genes, non-genes, it can also predict promoters. Promoters melt at a lower temperature than the gene protein coding region. Thermodynamically, promoter seems to be less stable than the protein coding region. That's interesting. Similarly, intron exons, this is a rice genome. And what we find is that these introns, which are identified as the black ones, this is Oryza sativa, technical name for rice genome, right? And peaks are the exons, and the valleys here are the introns here. So intron exon detection is not that easy, but this ChemGenome is able to identify. That's the interesting part. And what we find is when you scan about 7.6 billion RNA genes, what you interestingly find is that each of these categories, mRNA, tRNA, ribosomal RNA, right, and microRNA, they all have separate parts, separate areas on this stacking hydrogen bond salvation kind of a 3-dimensional plot. So that's the interesting segregation. So essentially tells you that maybe DNA wants to talk through its physicochemical properties, right? So that's the underlying message that's coming out. More interestingly, transcriptional start sites, promoter sites, and they don't have a conserved sequence. Typically, promoters, we have consensus sequences, but the deviations are far too many. Like if you see, all of these sequences are doing the same job, right? And then if you say same color across vertically, then it's conserved. But as you find, there is no such column at all. No column has one single color, right? There seems to be a lot of promiscuity, as they call it, right? So because of this, promoter detection is not easy from training point of view. If you have to train, which one would you train, right? But suppose you go to structural energetic properties of DNA, like I was mentioning, hydrogen bond stacking, solvation. So these are energy properties. What are the structure properties? Structure properties, base pair, tilt, twist, and those dihedral values that I mentioned to you. So you take all those 31 parameters, and each of the parameters shows a clear signal at the start site, transcriptional start site. So that's the interesting part, that DNA does want to talk through its structure, through its energy, right? And you don't need to worry too much about training. And similarly for the intron exon. Intron exon is very difficult to detect because there are only 2 sequences on either side of the intron exon. And the structural properties are able to show clear signals, right? So this came out last year on the intron exon part, right? So it's still evolving. I have my student now has a intron exon characterization software ready to be submitted. So we're trying to working on it. So essentially what I'm summarizing here is that each of the genomes at the NCBI is like a nice novel. We still don't know the language of DNA, but someday soon we'll be able to figure out and understand human genome then is like a Harry Potter novel, Rice Genome 2, Buffalo Genome 3, etc. without having to worry about this heavy-duty computation. So let's go beyond the computations to be able to read. Like the English language, 26 letters is not difficult at all, but 4 letters are causing us problem. We should overcome this problem using some universal language, right? Now, on the drug design discovery part, most of the drugs are targeting proteins. 90% of the marketed drugs target proteins. So from DNA, you identify, or RNA, you identify the gene. From there, you have a protein. And there is a gap between structures which are available in the protein data bank and the experimental sequences. And this AlphaFold is trying to get the structures of all of them. And people are sometimes questioning whether the structures are accurate or not, and so on. But that is a different story altogether. But there is a gap between experimental sequences and experimental structure. When I say experimental, X-ray, NMR, cryo-electron microscopy, etc., those kind of structures. And you need a structure to do a structure-based drug discovery, right? This is an earlier story. Now, again, this AI has overtaken this problem. AI has solved this problem, but experimental structures are few in number, or most of this if you want to do drug discovery. So what is the problem? Problem is generating a structure. And that boils down to, if you're given a sequence R, here R, etc., is an amino acids, different amino acids. If you're able to specify phi-psi, Ramachandran angles, then you have nearly solved the structure. When I say nearly solved, hello, are you able to hear Vijay Bhargav, ma'am? So everything okay?
Yeah, it's okay, sir.
Can I continue?
My next lab, all the diagrams of the special senses and the theory part should be done.
Yeah. Can I continue?
Continue, sir.
Okay.
Yeah.
All right.
Date,
So protein. So we call this as a
number.
protein. I think there is some noise. I think there is some class noises going on.
Yeah, Fatima. you please off your mic. Yeah. Okay, sir.
Oh, thank you very much. So, coming to the physicochemical aspects of protein folding, forget the prediction, structure prediction part. So, what we believe is that folded protein happens to be at the bottom of the funnel, the free energy funnel. That is the Anfinson's thermodynamic hypothesis, right? And it continues to be a grand challenge from the physics and chemistry and biophysics point of view, not so much from the ML AI point of view. Right, so we created a Bhagirath pathway, which is force field based by force field physics based methodologies. Right, so we do generate several structures and we compute the energies and calculate the free energies and so on. So for small proteins, that was an easy problem. So this was way back in 2005, and then we created a nice server here, web server, Bhagirath web server, which people can give the sequence and get a structure. So this was 2006. But beyond that, how to go to bigger proteins. So that was the motivation to add homology into this ab initio part. So that now enabled us to tackle proteins of any size, right? So, but there are some interesting things I would like to share with you on the proteins part, right? What we see is that from physics and chemistry point of view, protein folding is unsolved. Protein DNA unsolved, protein RNA unsolved, protein unsolved. Protein ligand, small molecule, that's a drug discovery, right? That drug discovery is also unsolved. Like you don't have routinely molecules. If you have a protein, molecules cannot be predicted just by looking at them, right? So that is an unsolved problem. So it's essentially a recognition problem. Small molecule or a big molecule, etc. Recognition involving proteins. So something must be wrong with the understanding of the proteins. Why are we going wrong? So that brings us to the question of, do we really know the language of proteins? Right? We know there are 20 amino acids. We have classified them into several categories: aliphatic, hydroxyl, aromatic, acidic, basic, sulfur, etc. But with all these classifications, we are nowhere, right, in terms of understanding protein folding issue. So sequence to structure, this Anfinsen hypothesis, yes, we know. And the secondary structures, Pauling's story, alpha helices and beta sheets are formed. That also we know, structures have verified them. So this is a Nobel. Right, and remember, he's one of the other guys who got 2 Nobel, right, out of the 5, right? And then Hausmann's idea that polar residues are outside facing water, meaning aspartate, glutamate, and aspargine, glutamate, etc. Charge residues and polar residues. Non-polar residues like valine, isoleucine, etc. They are buried inside. This is our extension of a thermodynamic hypothesis. He said oil and water don't mix, but we are extending it to structure. Right? This is our idea. Ramachandran said, out of these phi-psi possibilities, you have 180 degrees here, 360 here, 360 here. Out of all these possibilities, only 15% is occupied. Rest of it is not occupied due to steric clashes, right? So in spite of all these advances, we are not able to generate a 3D structure routinely, all right? Just like we do for DNA, secondary structure, double helical structure, we're not able to do it for proteins. So the one key we suggested at that time, so this was back in a few years ago, my student Dibara did this higher order Ramachandran map. Go to not just phi-psi, add these neighbors as well. add neighbors of phi 1, psi 1, phi 2, psi 2, phi 3, psi 3, then your space starts to shrink from 15 to allowed region will be less than 2%. Less than 2% allowed region will mean you are able to create a 3D structure. So that was the idea. One more interesting thing about the proteins is that when you analyze this from a mathematical perspective, you have, let us say, four colors and three sides to a triangle here. And you have 64 possible, 4 times 3, 4 times here, 4 colors here, 4 colors here, 4 colors here, 1, 2, 3, 64 triangles. But because of the because of the symmetry of the triangle, you may end up with just 20 unique triangles. But you may say this has nothing to do with proteins, right? Obviously, yes, right? But let's examine this further. Red color occurs only 10 times in these 20 triangles. Blue occurs only 10 times. Purple, green, 10 times each. Red and blue only 4 times, etc. Red and purple only 4 times. Red, blue, green, there are only 3 sides, so only once. Now, if you say that proteins, amino acids have evolved and now they got optimized over the course of evolution, right? We have to make a hypothesis. Amino acids have evolved out of 4 basic properties. A minimum of 1, maximum of 3 properties are used. Each property occurs in exactly 10 amino acids. Any 2 properties can occur in 4, etc. This is your hypothesis. Does it work? Now look at any textbook. All the classifications that people have proposed about amino acids don't seem to obey the rules, meaning these rules are nonsense and the textbooks are okay, or textbooks are imperfect, right? They're not perfect and your rules are okay, right? Let us examine this further. Is there any property which occurs only 10 times in an amino acid? We find that sp3 hybrid has gamma carbon. Some of you who have basic chemistry done, 12th class chemistry, you know what is a gamma carbon, right? sp3 gamma carbon. Ethane has gamma, sp3. Ethylene has sp2. Acetylene has sp. Remember that? So gamma carbon is main chain has alpha carbon, side chain, C beta, then C gamma. Glycine doesn't have a beta. Alanine has a beta. Valine has a gamma, etc. So exactly 10 amino acids have a sp3 gamma carbon. Hydrogen bond vulnerability, again exactly 10 amino acids. What about these two together? They can occur only 4 times, right? Green and blue or green and purple can occur only 4 times together. In fact, it so happens that these two occur only 4 times in K, Q, R, T, etc. So we have 4 properties identified and all of them seem to satisfy the triangle rule, meaning that people are not looking at the proteins correctly, and these are never mentioned in any textbook, biochemistry textbook, that these are the properties which are relevant for proteins. In fact, when we did homology modeling using just these 4 properties, people use thousands of properties. You can push the homology models to a very high level of accuracy, way beyond. Some of you may have used a Swiss model, etc. These are way beyond what you can achieve. So that's interesting, right? And then coming back to evaluation by peers, right? So this is a global competition. We call this CASP, CASP, and this is CASP 12 story. Bhagirath, of course, was for a low resolution structure. Bhagirath was doing very well. For a high resolution structure, meaning if you want a 3 angstrom RMSD from native, we are dropping a little bit in terms of our ranking at that time, right? And then 2018 was, they ranked us at the 13th position. Earlier it was 11th position. So we were examining as to why not number 1, why not 12, 13, etc. For low resolution, we were doing fairly well in the first bunch, right? So that gave us some motivation to improve our methodology. Now what happened during 2020 was the interesting part. AlphaFold, Google announced AlphaFold and everybody adopted this AlphaFold code. So they stopped doing their own research. So we decided, let's not do that. Let's just continue with our physicochemical methods. And how does it compare with AlphaFold? Well, we find that out of 49 on the 94 targets which were released, 50%, more than half of them are within 3 angstroms of AlphaFold. So I would say this is a success story for physicochemical methods, whereas you know what forces are governing this structure. Right? These are two structures. I'm just showing you 2 structures here, blue versus red, AlphaFold versus Bhagirath structures. These are all above 1,000 amino acids.
మన దగ్గర ఏమైనా డిస్కనెక్ట్ అయిందా సాజిదా
Yes, ma'am. Yes, ma'am. Yes, ma'am. It's from
ఎట్
our
అవర్
side.
ప్లేసా
Yes, ma'am. We are trying to reconnect, ma'am. Oh shit. How is it?
ఇన్ఫార్మ్ టు సార్
Yes, ma'am. Yes, ma'am.
ప్లీజ్ ఇన్ఫార్మ్ హిమ్
Okay, ma'am.
Today is not my day of technology, wise looks like, right? Anyway, so Dr. Vijayabarg, you are able to see my serious issue here. So now I'm back. I'll wrap it up in another 5 minutes. Hello? Are you able to hear me? Yeah, just wrap it up in. I'll just close it very soon now. Looks like there's some technical issues coming up.
yeah
All right, so coming back to the idea of the protein structure, you need some magic, so hopefully we are working on it. Now back to our Sanjeevani methodologies. There is an echo here somewhere. Okay. I'm trying to see if we can prevent the echo here. Yeah. Okay, maybe now it should be all right. Are you able to see my screen?
no sir no 1 minute ఏమ్మా
Yeah, today is no.
సాజిదా is there any issue
Yes, ma'am. We all have to mute our mics, ma'am.
Slides.
Sir, we are not able to see the slides, sir.
Sir, one sec.
I think Chair, sir is not. Sir, please, you can unmute now. So you can unmute now, sir. So please unmute yourself, sir. Jairam, sir.
Sir.
Yeah, I'm unmuted. Right. Can you hear me now?
Yes, sir.
I think I'll looks like I better wrap up shortly.
So we are not able to see your presentation.
Yeah, well, I'm doing that right now. Has it come
Yes,
up
sir.
now?
Yes.
So I'll go to this slide where I left off. I'm now focusing on only the third part, which is the Sanjeevani part. I'll mention to you the softwares that we have, which are freely accessible for some of you who may be interested in doing some molecules. I'm sure some of the faculty who are participating may already have a molecule or have a target in mind. They want to do some research. And how do we go about it? So leave the protein folding and all the challenges, etc. But so now the question is one of this, Sanjeevani, how do we use these softwares? So this software, Lipinski rule of 5, tells you about bioavailability, and it's easily available if you go to this utility. In fact, it's a very popular software. A lot of people submit their molecule and want to know whether it is Lipinski compliant or not. So this one. Then another is a protein part. Suppose you have a protein and you don't know the active site. So this will enable you to get you, it will suggest 5 binding pockets, and you can do an automatic docking as well of your molecule. You can upload your molecule and the protein target, and then you have this adds. So this is some accuracy comparison with other existing software in terms of active site, in terms of docking accuracies. This, I would say, is a very interesting, useful tool, RSPD tool. What it can do based on these physicochemical descriptors is to screen millions of compounds in a matter of minutes. So if you have a big library, let us say you want to do zinc database, or you want to do all the Ayurvedic medicinal plant and medicinal plant molecules, right, against your target. You can do this in a matter of minutes. So that's the interesting, useful thing. Now, 2024, we have it RSPD Plus, which is augmented by the machine learning methodologies, random forest, etc. So it works quite well. Then this BioTalk is an interesting tool which gives you the bioactivity information of an organic compound. Sometimes, we know that there is some activity, but we don't know exactly which protein is it binding to or which disease it can cure. So this tools enables you to do that. So this is a charge derivation tool. Paradoc is a docking software, blind docking accuracies. Here I'm showing you some delta G versus experimental part. This was a earlier version BAPL. Now the latest BAPL Plus, again, which is augmented with ML, does very well. So we have this BAPL Z, Paradoc, etc., and the sites of metabolism, DNA-based docking, minor groove versus intercalation. So such softwares are already available at the SAF Bio site. They're published and they seem to work quite well. So we know using these computational methodologies can do quick affinity versus specificity type matrix, off-target binding in particular. Suppose you're worried that the drum molecule you're suggesting binds to target 1, but probably does it bind to other targets in the human proteome, for instance, if there are 20,000 proteins. You can do that kind of a docking to see whether it binds. For example, this drug 14 is binding to target 1, it is binding to target 9. So this off-target binding, which can cause side effects, right? So you want to eliminate it beforehand. So you're taking care of this toxicity arising out of off-target binding. Such things can be done on a computer itself. You don't need to go to the test tube to do this. So success stories of Sanjeevani are, we have some anti-cancer molecules. So when I say success, it means you have done a prediction and then you're doing a testing in a laboratory, experimental compounds. So this was in collaboration with MIT School, Seema Bhatnagar from MIT, she's a professor in biotechnology. So she did the validation part, experimental validation. Then I already mentioned to you HBV part. So I'm not mentioning HBV here again. So this anti-Alzheimer's acetylcholinesterase, and again, the molecule predicted seems to work quite well experimentally, showing micromolar, back to low micromolar activities, right? And that is done through collaboration with Jamia, Milia, and Sanjeevani again, and methyltransfer anti-malarial. This is also through Jamia Milia. So this is a protein. This is from our School of Biological Sciences. So we did some antivirals against P. coronavirus, and that got published a few years ago in virology as well as web journal. So that's a good news. Of course, one of the molecules that we service, and my collaborator Hari Babu from Harvard Medical School enabled this. That's a good news. Sanjeevani has entered Nature a few years ago. So we now have, of course, a mobile app for Sanjeevani. I'm sure everybody wants a mobile app these days, right? So what is missing in drug discovery? Why is it that we cannot get a molecule end of the day, 10 a.m. to 5 p.m.? We are still working on the T part, the toxicity part. If you can take care of the toxicity and come up with some 5 rules of toxicity, you'll benefit mankind and drug discovery in a big way. We have some ideas of target binding, metabolism, related toxicity, etc. But biology of toxicity is still like a black box. If you can identify the steps, then you can easily write a software, a chemistry-based software, etc. So if you could do something about T part, right now some people are using machine learning, but there's not enough data to train on toxicity which molecules are toxic to humans, etc. That kind of information is not available in large quantities for trainings or machine learning methods to be successful. So I think I'll stop here. I've taken a lot of your time with these technical glitches. I'm sorry about it. So thank you very much for your attention. Madam, so mic is yours.
Yeah. Thank you, sir. I think it is giving the inspiration for the participants that they can do their works at their places.
Yeah.
Yeah. I think they can use it individually.
Yeah, if they have any questions, they can always mail them to me, right? They have some difficulty in using a software, they can always send an email to me, and I'm sure I will take care of it.
Yeah.
No issue. Right.
It's very inspiring talk, sir. Really, see, everything is possible, but we didn't know that unless we attended your talk today. Yeah.
Yeah, thank you, ma'am. Thank you very much. Wish you all the best with your remaining proceedings. I think you have a lot of lectures packed already.
Yeah.
A lot of hands-on session.
Thank you, sir. Thank you so much.
Good luck to you, and good luck to all the participants. All the best. So I will sign out now. So
yeah.
Thank you, sir. Thank you for your presentation, sir. We are highly inspired, and we will surely apply all the knowledge we have gained from you.
Good luck to you. Thank you very much.
Practically try to use ChemGenome, Bhagirath, and Sanjeevani.
And with a request to go beyond the existing software, right? Create new knowledge. I will encourage you to create new knowledge and new molecules that benefit the world.
Yes, so we will try to understand the DNA language for sure.
Wonderful. All right. Take care. Take care. Good luck to you. Bye now.
Thank you. I request all of you to fill the feedback form posted in the chat box and also in the groups.